Okay, so we'll start with our next topic, that is biomolecules. As you can see, the first question arises here is, what are biomolecules? So, any form of living system, be it a plant or be it an animal, the molecules which is found in it, that is carbohydrate, proteins, lipids, are considered to be molecules. We have already seen that the membrane, that is the plasma membrane, has proteins, carbohydrates, and lipids, and there are many more things. These all are considered to be biomolecules. Okay, so biomolecules are organic as well as inorganic chemical compounds that form the structural and functional basis of living organisms. Okay, they participate in essential life processes including heredity, cellular structure, and energy storage. What are those molecules? Carbohydrates, proteins, lipids, nucleic acids are the four primary categories. Okay, so four primary categories of biomolecules, that is carbohydrate, protein, lipid, and nucleic acids. Okay, so next we'll move on to the elements which are found in the earth and the elements which are found in the human body. A comparative approach. Okay, in order to understand which form of element is existing in abundance. Okay, if you see in the earth crust, 46% of the earth crust is primarily oxygen, then silicon. And if you see a human body, it's again oxygen but carbon. Okay, what it denotes is carbon being the part of the organic compounds, that is, it is the substance which is found in every type of living systems. Okay, so this is what it suggests. I'll show you it in a tabular form. Saringla, see, if you see clearly, silicon and oxygen are about 46 to 27% on the earth's crust, and if you compare it with the human body, that is the living matter, it is uh, carbon, if you see, it's 18.5% and oxygen being 65.0%. Okay, very important things, questions may be asked. Okay, okay, now we'll move on to the next, chemical analysis of tissues. How do we say that some molecules, that is molecules, are again categorized into different forms based on some chemical analysis tissues, chemical analysis method? What that method is, trichloroacetic acid. What is trichloroacetic acid? Cl3CCOOH. Okay, what we do is we take any form of a tissue, be it a plant tissue or an animal tissue, we grind it in this acid, it is trichloroacetic acid. When we grind and when we filter it through a cotton or whatever, what we get is we get a soluble part, acid soluble part, and an acid insoluble part. Now this are categorized again into macro and micro molecules. Okay, we'll deal with it. So tissues are homogenized and treated with trichloroacetic acid, which precipitates macromolecules while keeping small biomolecules soluble. So which are considered to be insoluble? Macromolecules or micromolecules? Insoluble part is the macro form of the molecules. Okay, it is considered to be retentate. It is called retentate. When you filter it, something which do not dissolve in the acid remains on that filter is considered to be the insoluble part, and it is known as the retentate, and it is considered to be macromolecule, and which is dissolved in the acid is considered to be the micro form of the molecules. Okay, so this is why this method is done, in order to find which is a macro and which is a macro. The another form of test which is common in characterizing biomolecules is the ash test. Okay, what do we do is tissue samples are completely burned, leaving behind inorganic residues. When a particular tissue is burnt, all the organic content remains or escapes in the form of gases. The black ash which remains actually tells us what? The inorganic part. What is the inorganic part? The minerals which are present in the particular tissue. Okay, so what was the result of chemical analysis? You got acid soluble fraction which contained what? Note this down. Amino acid, nucleotide, sugars. Sugars means simple sugars. That is monosaccharides. Okay. Not all form of carbohydrate is considered to be a macromolecule. Monosaccharides are considered to be a micromolecule. Okay. And some inorganic ions, they generally have their molecular weight less than 800 Dalton. Okay, this is also, this acid soluble fraction is also known as the filtrate. Okay, filtrate. And what is the acid insoluble fraction? It is known to be the retentate. Okay, this includes macromolecules in the form of proteins, nucleic acid, polysaccharides. See the name clearly. Polysaccharide is again a form of a carbohydrate, but it is a complex carbohydrate. Okay, done. Sorry. So this is what is the result of chemical analysis. There are definitely some significances. Okay. So what are the significance? The separation provides insight into biochemical diversity of cells between small functional units and larger structural biomolecules. Different forms of molecules are found out by using this method. Done. Now we shall move on to the difference between a micromolecule and a macromolecule. So as we discussed, micromolecules are small and simple molecules which are having a molecular weight less than 800 Daltons. It also includes amino acids. Did you note amino acids? Sugars, nucleotides, vitamins, as well as inorganic ions. Okay, and if you consider macromolecule, they are large and complex. They include protein. See, amino acid and proteins are connected. If an amino acid is found in a pure form, not in the form of a chain, but a pure amino acid, a single amino acid, it is considered to be a micromolecule. But when it is in the form of a chain, continuous chain, it is considered to be a protein and is a macromolecule. Okay. If you see in macromolecules, one thing is written as lipid, but there is an exception for lipid. Okay, it actually does not has a weight more than 800 Daltons. Okay, it does not has a weight more than 800, even though it is considered a macromolecule only for the reason because it is insoluble. Okay, acid insoluble. That is why the lipid part is considered to be what? A macromolecule. But it is actually less than 800 Daltons in molecular weight. Okay, that was an important point. And nucleic acid, when it was just a nucleotide, that is when it was either adenine or guanine or thymine or cytosine, Singly it was considered what? A pure form of a micromolecule. But when it is a nucleic acid, that is either DNA or RNA, it is considered to be a macromolecule. Very important part. Okay. So micromolecules are actually the building blocks of the macromolecules in a way, right? The micromolecule being the amino acid is the building block of the proteins. Hmm? Nucleotides is the micromolecule which is the building block of the nucleic acid. So now we'll move on to the next. That is again one point that we just left was the macromolecules. Their molecular weight is higher, and it is also about 10,000 daltons. Okay, and this is between 8 and 800 daltons, less than 800 daltons. Okay, here the exception is lipids, which is less than 800 daltons. Okay, is it clear? Shall we move to the next? Okay, so again the difference between the organic and the inorganic part. Organic means the carbon based molecule, you can say carbon based molecule, but what is CO2? This is also having carbon, but is it organic? No, it's an inorganic part. So it not necessarily means that carbon, if it is present, it is considered organic. Carbon dioxide is actually a inorganic molecule, as you can see here. Okay, so carbohydrate, protein, all those things which we have already seen. Inorganic are non-carbon based, and the other point for organic is that it should be the part of a living system. Okay, living system. Okay, now inorganic biomolecules, water, minerals, salts, and gases. For example, oxygen and carbon dioxide. Okay, but life does not only depend on the organic part; it depends on both. Okay. Shall we move to the next? Take time. Take time to note. We will move to the next topic. So, organic and inorganic we have seen already. Now, the other important thing here which we have to discuss is primary versus the secondary metabolites. Very important. Okay. So, what is a primary metabolite? These are substances which are produced by the living system, which is going to be used in some metabolic process, any chemical reaction within the body. This is going to be utilized, essential for basic cellular functions like growth and development. Key term. Okay, example amino acid, nucleotides, simple sugar, organic acid. But if you consider secondary metabolites, these are produced by the body only, but is not actually used. Okay, these are not directly required for survival, but play ecological roles. Play ecological roles such as defense, signaling, competition. Okay, suppose there is a plant tissue, a host plant tissue. It is infected by a pathogen. Okay, what this host will do is the region of infection, wherever the infection is caused, host will produce some substance, some toxins. What? Some toxins. What will eventually happen? The pathogen which is at the region of infection will eventually khatam, die. Okay. So this is what is secondary metabolite. Okay. No direct requirement by the body, indirect requirement in the form of defense, signaling, competition. Examples like alkaloids, flavonoids, antibiotics, pigments. We'll be dealing with them particularly one by one. Okay, don't worry. So now we'll move on to secondary metabolites. Okay, shall we? Hmm. Secondary metabolites, we just saw that as per NCRT, the types of secondary metabolites which are listed is alkaloid, terpenoid, phenolics, and some form of other secondary metabolites. We'll go one by one. What is an alkaloid is? Alkaloid is a, see the important word. It is a nitrogen containing secondary metabolite and is only derived in plant. Animal tissue cannot have alkaloid, okay? Only specific for plant. They often have strong physiological effects on humans as well as animals. Okay, so we just saw that secondary metabolites are not directly used for the that organism which it is produced, but they can be used for what then? They can be used for human welfare. Okay, for the human welfare, these alkaloids are used. Example, if you see examples of alkaloids, I've listed it here. Very important. Morphine. Morphine is an analgesic. What is an analgesic? Which reduces pain by acting on the neural system or the nervous system. Okay. Quinine. It is an anti-malarial alkaloid. Codeine, cough suppressant. Caffeine, stimulant. Okay. We drink tea, we drink coffee. What those things contain? Caffeine. Okay. It is a form of a stimulant. Nicotine, again a stimulant. What are those things which are considered stimulants? Which we think that we have to take it again and again and again and again. Okay, these are considered to be stimulants. For example, some people used to smoke or they take tobacco due to this substance, nicotine. Okay, as well as strychnine is a neurotoxin. Okay, so these are examples of the alkaloids. Okay, they are widely used in medicine as painkillers, anti-malarial stimulants, muscle relaxants. Okay, done. And within the plant system, they act as defense compounds within the plant, which is producing actually the alkaloid. In those plants, these alkaloids act as defense. Okay, defense compounds. Clear? So now we'll move on to the next, that is terpenoids. Okay, if you see, terpenoids are derived from isoprene units, chemical unit. Okay, you have to memorize these. So it is a large class of, not a single class, large class. It contains many subtypes, it means. Okay, isoprene units. Okay, they are often volatile. Volatile means gets easily evaporated, right? So volatile means evaporates easily. Again, they are aromatic. They contain benzene ring. Biologically active. They contain what? Large class of what units? Isoprene units. Okay. Examples: rubber. Okay, it is a polyisoprene. Carotenoids, which is a pigment in plants, right? Essential oils like eucalyptus oil, citronella oil, menthol, camphor. All these are considered to be what? Terpenoids. Okay, actually used for human welfare again. Okay, so used in medicine. Camphor is used as antiseptic. Menthol used as a cooling agent. Nowadays, soaps also contains this particular compound. Perfumery. Okay, normally in perfumes, volatile liquids are used, and it is an example. Okay, pigments in order to dye certain form of a fabric. This also could be used. Okay, and industrially a natural form of a rubber. Okay, so these are nothing but terpenoids. Okay, so now we'll move on to the next, that is the phenolics. Okay, phenolics. What are these phenolics? These are again aromatic and they are characterized by the presence of hydroxyl groups. Hydroxyl group means what? Alcohol group, which is attached to a aromatic ring. Aromatic ring we have seen in chemistry, organic chemistry. What this compound is? Phenol. That is why phenolics. Okay, and again they play protective role in plants, protective as well as structural roles in plants. Okay, phenolics are phenolic compound. This is the benzene ring. This is the alcohol group. Okay. Okay. What are the examples? Tannins, which is used in the plant defense. Lignin, it is a polymer. We have seen the secondary cell wall, or if we consider the sclerenchyma tissues within the plant, is composed of what type of cell wall material? Lignin, right? So lignin again in some parts contain these phenolics. Resins, okay, protective exudates, okay, liquid type of substance which are sticky in nature, okay, and they do not have color, colorless compounds. These are considered to be resins, okay. These are again what? Phenolics. Tannins are used in what? Leather industry, by in the process of tanning, right? Okay, it is used in medicine. Lignin provides mechanical strength to plants and industrially used in paper, okay. Resins is used in varnishing and perfumery. So those wooden surfaces, they tend to appear new always because it is tarnished using what? Resins, okay. So this is what is secondary metabolite. Okay, you can take time to note, then we'll move on. Okay, I hope you have written the content. Now we'll move on to the next. So other forms of secondary metabolites, as you can see, these are nothing but antibiotics, pigments, and essential oils. Antibiotics are those substances which are actually produced by the microbes. Okay. Secondary, they are again secondary metabolites. For example, penicillin from penicillium, streptomycin from streptomyces. Okay, this is a form of a bacteria, and penicillium is a fungus. Okay, the compound which is extracted is important here, not the organism itself. Okay, so pigments include carotenoids, anthocyanins, flavonoids, which provide color to the plant. Pigments are also considered to be a form of a secondary metabolite. Okay, essential oils, volatile aromatic compounds like eucalyptus oil, which we have already discussed, clove oil, citronella, which are actually used in perfumery, flavoring, in traditional medicine. Okay, so these are considered to be what? Again, secondary metabolites. Okay, Okay, so now we'll move on to the next topic, that is carbohydrates. Okay, so we know that carbohydrates are considered to be the hydrates of carbon. These are considered to be the hydrate de carbon. It's written in French. It means hydrates of carbon. Okay. It has a general formula of CnH2ON. Okay. So, uh, what is the function of the carbohydrate? It is these are very basics. Okay. We'll just revise them one by one. Source of energy, then precursor for many organic compounds, that is the fat, the amino acid. Precursor, the primitive form, is a carbohydrate. In the membrane we have discussed that carbohydrate is seen in the form of glycoprotein and glycolipid. That is what is written here. Okay, and it is the component, structural component of plants. It is the cell wall material. Primary cell wall is composed of cellulose. So these are those basic things which carbohydrate is responsible for. Okay. Now this carbohydrate is classified again based on its subunits. Okay. So if we consider it, the classification, the carbohydrate is categorized into three: the monosaccharide, the oligosaccharide, and the polysaccharide. Right? What is a monosaccharide? Is the basic unit of the carbohydrate which cannot be further hydrolyzed. Hydrolyzed means broken down by the action of water. Okay, so it cannot be further hydrolyzed. Very important point regarding monosaccharides. Okay, now based on the number of carbon atom, it is again subdivided, and based on the type of functional group, again it is subdivided. What? Monosaccharides. This will be dealing later in this session, but this is one of the type of the carbohydrate. Now if we consider oligosaccharide, this oligosaccharide can be further hydrolyzed, that to 2, 3 or 4 units. 2 units, it can be either hydrolyzed into 2 units, or hydrolyzed into 3 units, hydrolyzed into 4 units. Based on the hydrolysis and the number of units which it is giving, it is categorized again into many types. If it is giving 2 subunits, it is considered to be a disaccharide, 3 trisaccharide, 4 tetrasaccharide. Easy? Okay. What is a polysaccharide? Non-crystalline, non-soluble in water. These were actually soluble in water. These both were soluble in water. But polysaccharides are not at all considered to be soluble. They are insoluble. Okay, and they do not have taste. These are sweet in nature. Okay, generally these are sweet in nature, and these are actually considered sugars, which we saw in the form of simple sugars. Now you just tell me whether this is a micromolecule or a macromolecule, that is the mono and the oligo together. Micro or they are considered to be the micromolecule. Okay, which are soluble or insoluble in acid, trichloroacetic acid. Soluble, right? Soluble part, acid soluble part, and this is actually the complex, not sugar but carbohydrate. Okay, so carbohydrates again categorized into two: micromolecule and macromolecule. Micromolecule consists of monosaccharide and oligosaccharide, and polysaccharide is considered to be a macromolecule. Okay, they, on hydrolysis, they give number of monosaccharide, not just one. They are a polymer of monosaccharides. Polymer means a continuous chain. Okay, examples are starch, cellulose. Okay, Now we'll move on to, wait, we'll move on to the, um, just a minute, let me just change the slide. Monosaccharides. Again, monosaccharide is categorized based on the number of carbon atoms I told, right? So if you see, based on the number of carbon atoms, it is of many types: trioses, tetroses, pentoses, hexoses, heptoses. What is a triose? Triose means it has how many carbon atoms? See, 3 carbon atoms. Number of carbon is 3. Example is glyceraldehyde or dihydroxyacetone. Both are what? Both are trioses, but are they having the same functional group? No. We'll be dealing with it in this part of the slide, okay? So based on the number of carbon atoms, trioses, tetroses. For the example of tetrose is erythrose, triose. Pentose example is ribulose, cellulose, arabinose. Okay. And there is also an example called ribose. Ribose sugar is the component of what? Nucleic acid, right? Nucleic acid, that is DNA or RNA. This we already know. Okay. Now hexoses. Hexose consists of 6 carbons. Has glucose, fructose, galactose, mannose. Okay. And heptoses, pseudoheptulose, glucoheptose. Okay. These are the forms of monosaccharide based on the number of carbon atoms. Whether they are hydrolyzable, can they be hydrolyzed? No, they cannot be hydrolyzed further. If you try to hydrolyze a glucose, it will not break into 6 carbon atoms by the action of water, right? So it cannot be hydrolyzed further. So now based on the type of the functional group, basically in monosaccharide, only 2 forms of functional group is seen. Alcohol is also seen, but what makes them different is 2 forms of groups. That is, one is the CHO group, which is considered to be the aldehyde group, and the other is the C double bond O, that is the ketone group, right? Okay. If it is containing, what is this at the end? The first carbon is containing CHO, right? CHO means what? Aldehyde. So the carbon compound, which is a monosaccharide, if it is having a aldehyde group, it is considered to be a aldose. What? Aldose. Aldehyde in the aldose. Okay. But if you see in the second carbon over here, this is having a ketone group, and this will be considered as ketosis. Okay. What is the example of aldoses? Glyceraldehyde. Aldehyde, which was how many carbon? 3 carbon, based on the number of carbon atoms. But based on the type of functional group, it is glyceraldehyde. Again, glucose. Glucose is what? Having 6 carbon atoms, but is a aldose. Same number of carbon atoms is seen in fructose, but fructose is not an aldose. It is a ketose. Okay, see, it is fructose. This is a fructose molecule, and this is a glucose molecule. See, 6 carbon here also, 1, 2, 3, 4, 5 and 6. 6 carbon. Both are the same number of carbon atoms, but based on the functional group they are different, with different chemical properties. Okay, this is containing a aldehyde group, this is containing a ketone group. Okay, this is an aldose, this is a ketose, but based on the number of carbon atoms they are of the same category, that is hexose. Okay, I hope this is done. You can have a note. Okay. me to write the next. Okay, do it soon. We just discussed galactose, mannose, glucose, and fructose, right? Mannose, galactose are actually considered to be what? Epimers of glucose. Now, what is epimer, right? Galactose, mannose, these are considered to be the epimers of glucose. Okay, what is an epimer is isomer which forms by the interchange of the functional groups. Which functional groups? OH group and the H group. Which carbon atoms on the second carbon atom or on the third carbon atom or on the fourth carbon atom? What? I'll just note it down. Okay. So, epimers. These are isomers of a compound which differ in the arrangement of the functional groups OH and H, the OH group and the H group. Okay, at which position? At positions, that is the carbon atom's position. Okay, either at 2 or either at 3 or either at 4. Okay, we'll list out an example. An epimers of glucose. Okay, what did I just say? One is mannose and the other I told galactose. Okay, so mannose is an epimer which has this difference, the difference in OH and H. Okay, at which carbon? At the second carbon. Second carbon. Okay, and galactose is an epimer of glucose which is having this change in OH and H over the fourth carbon. Okay, when I say OH and H, what do I mean is this particular thing, H and OH. Okay, they reverse their positions. Okay, if it is reversing on the second carbon, then it is an epimer of glucose in which way? Mannose. If it is changing on the fourth carbon, this carbon, then it is very easy. Okay, just remember this. You can note this out. We'll move on to the next then. Then now we'll be dealing with some important. Uh, monosaccharides and their where they are found, what they are used for. Okay, some, this is actually asked in the form of match the following in NEET exam. Okay, so you have to memorize all these things. Some important monosaccharides. The first is ribulose, which is found in the nucleoplasm. What is the nucleoplasm? The matrix which is found inside the nucleus. Okay, so within the nucleus, this form of the monosaccharide is seen. Okay, next is arabinose. Arabinose is found in gums of plants like acacia. The plant name is itself Acacia arabia. What? Acacia arabia. And the other plant is Acacia. No, this is enough. Okay. It is found in the gums. Okay. Then, if you consider cellulose, it is a cell wall material. Cell wall in plants. Okay, a component of hemicellulose. Component of hemicellulose. Okay. Glucose and fructose we already know, but regarding fructose there is a very important point. That is the sweetest form of sugar. What? The sweetest form of sugar is nothing but fructose. Fructose is considered to be a sweetest form. But I asked sweetest. Now we are discussing regarding monosaccharide, right? And how you can say sucrose? Huh? Sucrose is not. Okay. Sweetest form is fructose. Okay. So it has the sweetest taste, and there is also some extra point regarding another form of a compound, which is again the sweetest form of a carbohydrate. Okay, it is considered to be thaumatin. Okay, thaumatin. Okay, which is actually extracted from a plant called thaumato. Not a plant; it's a bacteria. Okay, Thaumatococcus. You please note it down. Danieli. Okay, Thaumatococcus Danieli. The name seems weird, but you have to memorize it. Okay. Sorry. In the same way, aspartame. Aspartame is considered to be a artificial sweetener. Sweetener. Sweetest carbohydrate. Okay, sweetest form of carbohydrate. Noted? We have one thing left, that is mannose. Mannose we just discussed what it was, an epimer of glucose. Okay, it is found in the albumin of egg. Okay, albumin of egg, as well as it is found in the hemicellulose of plants. Hemicellulose of plants. Okay, and the last is rhamnose. Rhamnose, here the second carbon lacks lacks one oxygen atom. Okay, and this sugar is present in the phloem of plant, a conducting tissue. Okay, you can note these can be asked in the form of match the following in NEET exam. Okay, is it done? Okay, now we'll consider the next part, that is the derivatives of monosaccharides. What derivatives of monosaccharides? Monosaccharide might change its form, and it can be derivated into another form of a compound. Okay, so it is found in four forms basically: deoxy sugars, okay, deoxy sugar, amino sugar, sugar acid, sugar alcohol. Okay, if you consider the second, third, and the fourth one, you can easily find what is the derivation which has accumulated in these monosaccharides. In amino sugar, what is added upon? Amino acid, right? That is why it is known as amino sugar. In sugar acid, what is added in the monosaccharide? Acid, carboxylic acid, okay, or any form of an acid. Or in sugar alcohol, the functional group, that is OH, is accumulated. That is why these are considered to be derivatives of monosaccharides. What is a deoxy sugar? We know regarding deoxyribonucleic acid, right? It is found in what? DNA. That is, deoxyribonucleic acid is actually in DNA. Okay. What happens is, okay, so in the fifth carbon, no, in the second carbon, this is the second carbon. Okay, in the second carbon, usually there is H and OH. This is your ribose sugar, and this is the second carbon. Okay, if this OH, the oxygen atom which is here, is removed, what it becomes? What it becomes? Deoxyribose sugar. Easy. Okay, so this is the difference between an RNA and a DNA. Okay, very important point. This is again asked. It may be asked. Not very hard thing. Okay, sorry. So you can note this down. In deoxy sugars, deoxygenation of ribose produces deoxyribose, which we just saw, and it is a structural component of the DNA. Amino sugar, when one or more alcohol groups of monosaccharides are removed by amino groups, NH2, amine group. Okay, so the OH groups, not just one, it could be many, are replaced by amino groups, then that form of a monosaccharide is called as amino sugar. What is an example? Glucosamine. Okay, in the glucose, what happens? It alcohol gets replaced by the amine, which forms glucosamine is the component of chitin. Chitin is formed in the cell wall of fungus, as well as it is part of the exoskeleton of arthropods, for example cockroaches. Okay, fungal cellulose, as well as hyaluronic acid. Okay, hyaluronic acid. Sugar acid, what happens? The sugar acid, oxidation of aldehyde group to alcohol. Now here what happens is, the aldehyde group changes in aldehyde group, or the alcohol group forms sugar acids. Okay, example ascorbic acid. is a sugar acid, as well as sugar alcohols, reduction of aldoses or ketosis. Aldehyde group and ketone group gets converted into alcohol group, that is OH group. Example, glycerol and mannitol. Very easy, you can have a note of all these things. Then, okay. தம்பி எழுதியாச்சா? Okay. அடுத்தது போயிடலாம். Now, one more thing to consider here is, now monosaccharides, they can occur in both straight chain structure as well as in ring-like structure, right? So we'll see an example and we'll try to represent it, okay, diagrammatically or molecularly. So monosaccharides, straight chain as well as ring like structure, okay. Let us consider the example of glucose. You know that glucose contains how many? 6 carbon. So 1, 2, 3, 4, 5 and 6, okay. So the last carbon, that is the first carbon, is consisting what? The CHO group, okay, the aldehyde group. Then we have OH group, H group, HO, H, okay, H, H and H2, CH2, OH. So this is what? This is your glucose, right? This is the straight chain structure of glucose. It is also known as what projection? So this is your straight chain structure. The ring-like structure would form now this thing, this structure, ring-like structure, is very important because we are going to see the difference of alpha and beta isomers of glucose. In order to understand those, we have to memorize this ring structure, okay. So which is the first carbon? 1, 2, 3, 4, 5 and 6. Totally 6 carbons, right? Okay. So this is both the ring as well as the straight chain structure of your what? Which molecule? Glucose, okay. சரி. Fructose ல என்ன டிஃபரன்ஸ் இருக்கும் தம்பி? இதே ஆல்டிஹைட் குரூப் இருக்குமா அப்படின்னு கேட்டா இருக்காது. என்ன குரூப் இருக்கும்? கீட்டோன் குரூப் இருக்கும். எங்க இருக்கும்? ஃபர்ஸ்ட் கார்பனா செகண்ட் கார்பனா? செகண்ட் கார்பன், சரிங்களா? சரி. Okay, now we'll move on to understanding alpha versus beta forms, actually asked in the question paper, okay. So what is alpha glucose? In alpha glucose, the hydroxyl group at the anomeric carbon. That is which carbon? The first carbon. First carbon la irukra endha group? Hydroxyl group is positioned downward. Enga irukku? It is positioned downward. Okay. If the OH is facing downwards, it is considered to be what form? Alpha isomer. If that same hydroxyl group in the first carbon is positioned upward on the same side of CH2OH, then it is considered to be a beta carbon. புரிஞ்சுதா என்ன சொன்னேன்னு? இப்போ திருப்பியும் அந்த டயக்ராம் போகுமா? இப்போ போட்டது the diagram which we just draw, the ring structure. Let us now understand using the diagram, okay? That representation. இப்போ 1 minute. Now let us focus this diagram which I was talking about. Now the OH group is facing downwards or upwards? இது தாண்டா OH group, இது தாண்டா carbon 1. Downwards. Alpha or beta? Alpha. This is alpha form of D-glucose, okay? So this is alpha D-glucose. இதுவே இந்த OH வந்து எங்க இருந்திருக்கும்? மேல OH இருந்திருக்கும், கீழே என்ன இருந்திருக்கும்? H இருக்கும். இப்போ இந்த OH உம் CH2OH ஒரே சைடுல இருக்காங்களா? They are both on the same side, right? Now this is considered to be a alpha வா? இல்ல, நான் தலையாட்டுற beta D glucose. புரிஞ்சிருச்சுல? Very easy thing, but usually students do not know this, okay? Alpha versus beta. Alpha versus beta, what form of which monosaccharide we saw? D glucose, okay? So you can note this point. Alpha glucose la eppadi irukum? Downwards, opposite to CH2OH. Beta glucose la same side as CH2OH, upwards, okay? What is the biological importance? Alpha glucose polymerizes to form starch. Starch இருக்குல, உருளைக்கிழங்கு சாப்பிடுவோம்ல, potato is composed of starch. Starch சாப்பிட்டா டைஜஸ்ட் ஆகுமா இல்லையா? அடுத்த நாள் வெளியே வரும்ல, it comes out, right? It means it is digestible, okay? Whereas beta glucose polymerize ஆகுது, நிறைய யூனிட்ஸ் வந்து ஃபார்ம் ஆகுதுன்னா ஒரே ஸ்ட்ரெயிட் செயின்ல, அது நம்ம என்னன்னு சொல்லுவோம்னா we call it cellulose. Cellulose if we eat, whether it will digest, but animals like cows, in cows it gets digested, okay? So cellulose is indigestible in humans, whereas alpha glucose forming starch is easily digestible. Got it? Note this down. You want me to draw the diagram again? I will draw it again. No, we have seen it in the video, but while you are noting this, you have to draw it and practice. First you complete this, then I'll show the slide again and you can practice it out, okay? You have to remember this concept. Alpha D glucose, starch. Beta D glucose, cellulose. Alpha digestible, beta non-digestible, okay? Which carbon atom? First carbon atom, okay? It is considered to be what carbon? Anomeric carbon, okay? Monosaccharides follow which general formula? CnH2O in bracket and N. Okay, it follows that general formula. That general formula is not for the whole of carbohydrate. It's specifically for monosaccharides. Okay, carbohydrates are also known as the carbohydrates of carbon. Okay, you just note one more point. Fructose is also known as fruit sugar. Okay, fruit sugar. Glucose is known as grape sugar. We have not written anything regarding glucose. Write one more point. Glucose, also known as grape sugar, it is found in abundance in blood. Okay, and is also known as blood sugar. So it has two names, two general names. What are those? Grape sugars, blood sugar. And the mirror image of glucose is considered to be dextrose. Note this down, very important point. Okay, dextrose. What? Just it's not here, but you have to note this. Mirror image of glucose, that is its straight chain representation. If it is mirrored, the atoms get arranged oppositely, then it becomes what? Another form of hexose, that is dextrose. Okay, dextrose. Okay, note it down. Any doubts? So till now we have completed till monosaccharide, and it is 7:30. Okay. Now we'll move on to oligosaccharide. Okay. Let us complete this carbohydrate part. Okay. In the next part of biomolecules, we'll be discussing regarding amino acids and proteins. Now we'll complete carbohydrate. Okay. Sorry. So we have seen, and one more thing is the D and L forms of monosaccharide. So this is based on the optical activity. Okay. Dextro versus the levo form of the what? Glucose, specifically glucose. D-glucose means what? The hydroxyl group on the fifth carbon, that is considered to be the penultimate carbon. Okay, fifth carbon la இருக்கிற OH group என்ன ஆகும்னா right side la இருக்கும். எங்க இருக்கும்? On the right side. That is what is D form of the glucose. If the same OH group of the fifth carbon is on the which side? Left side. Then it is considered the see L left, L left, easily memorize. Sorry, enzymes are stereospecific. Most living organism use which form of sugars? D form of sugar. If it is using glucose enzyme, glucose use பண்ணதுனா எந்த glucose தான் யூஸ் பண்ணும்? எல்லா D ஆ? D glucose சரிங்களா? D sugars only the enzymes use. These are considered to be stereospecific. What is stereospecific? This D and L forms are nothing but considered to be stereoisomers. You have seen in organic chemistry, right? Stereoisomerism, same concept apply here. In stereoisomerism you also learn two type: geometrical isomerism, optical isomerism. This is a case of optical isomerism. Okay, and is seen in which type of carbon? That is not the sequence of the carbon. It is found in which nature of carbon? Chiral carbon, right? Asymmetric carbon. Chiral carbon la மட்டும் தான் என்ன தெரியும்னா ஸ்டீரியோ ஐசோமெரிசம் ஓட ஆப்டிகல் ஐசோமெரிசம் தெரியும். சரிங்களா? So chiral carbon or asymmetric carbon. It means what? It means that the fifth carbon in the glucose molecule is what in nature? Chiral in nature. Right side what form? D-glucose. Left side L-glucose. Mostly living organism use D-glucose for metabolism. The stereochemistry is crucial in biochemistry and medicine. Okay, note this stuff. I hope you have noted this. We'll move on to the next. Okay, D and L glucose is completed. Now we'll move on to oligosaccharides. What? Oligosaccharide. இதுக்கு முன்னாடி what did I say? I just said that it contains how many subunits upon hydrolysis? Either 2 subunits, 3 subunits, 4. If it is 2 subunits, disaccharide. 3 subunits, trisaccharide. 4 subunits, tetrasaccharide. This is what we say. So oligosaccharides are carbohydrates made up of 2 to 10 monosaccharides even, okay, which are connected by, those monosaccharide units are connected by what type of bonds? Glycosidic bonds. What bonds? Glycosidic bonds. Sucrose. Sucrose is a disaccharide of what type of monosaccharides? Glucose and fructose. Sucrose is what? Not a monosaccharide, disaccharide. It consists of glucose as well as fructose, connected by glycosidic bond. What type of glycosidic bond? Alpha 1, 2. Alpha 1, 2. This one is the first carbon of glucose. This 2 is the second carbon of the fructose. The first carbon of the glucose and the second carbon of the fructose get connected by the glycosidic bond. In the same way, lactose is a disaccharide. ரெண்டு monosaccharideால ஆனது lactose. என்னதுன்னா ஒரு பக்கம் glucose, the other side galactose. Okay, what bond? Beta 1, 4 bond. Very important topic, repeatedly asked. Okay, and can be asked again. Maltose, also known as malt sugar. Maltose is also known as malt sugar. Okay, not malt. Okay, now this is connected. It consists of 2 glucose. Rendume same type of monosaccharide, connected by alpha 1, 4 glycosidic bonds. Okay, glycosidic bonds are covalent type of linkages. We know covalent bond, we have studied in chemical bonding. Okay, and is formed between anomeric carbon. Anomeric carbon, which carbon I told? First carbon of one sugar and a hydroxyl group of the another. Okay, hydroxyl group of the another. By the release of H2O molecule, this glycosidic bond is formed. Carbon 1, carbon 2 in the case of sucrose. What happens eventually? நடுவுல இருக்கிற H2O molecule வந்து releases. Okay, that is why glycosidic bonds. Okay, so bond orientation alpha or beta determines digestibility and function. Which are considered to be digestible? Alpha or beta? Alpha. Beta is indigestible, as we have seen in cellulose. Okay, very important part. I think I have a diagram for the glycosidic bond. Wait, let us just see. If it is not there, I'll provide you through notes. Okay, don't worry. This is polysaccharide. No, I have not added it. Okay, don't worry. We'll see it in the next class. Time permits us. Okay, so we were here, right? Note it down. You have understood, right? Hmm? This is the basic definition. Okay. Now, separately, we can again see what is alpha, that is this types of glycosidic bonds separately for our better understanding. Okay. So we have seen all these things, glycosidic bonds and disaccharides. Disaccharides. This is- So alpha 1 comma 2, beta 1 comma 4, and alpha 1 comma 4. Again, same thing. Is it the same thing? So found in sucrose, glucose and fructose, the bond forms between the C1 of glucose, carbon 1 of glucose and carbon 2 of fructose by the removal of removing one H2O molecule. Okay, C1 and C2, C1 of glucose, C2 of தலையை மட்டும் ஆட்டுற நோட் பண்றா சரி beta 1 comma 4 bond in lactose. What happens? The bond links carbon 1 of galactose, which is in which orientation? Beta orientation. Okay, that is by beta. This was in which orientation? Alpha. The first, which is the first? Glucose is the first, right? It is in alpha. That is why this is called as alpha. Here the first is galactose, right? That is why beta orientation. Okay, and the C4 of the glucose. Galactose first, glucose next. Beta orientation. Okay, easy. And the alpha 1 comma 4 bond, which is found in the maltose sugar, it is also seen in starch and glycogen, which is a polysaccharide. It connects C1 of one glucose, which is an alpha orientation, of course, and C4 of another glucose. Easy, very easy thing. You can easily remember this, and this could be asked. Okay, please note it down, all the three, and we'll move on to the next. Okay, now we'll move on to the next thing, that is polysaccharides. Okay, what are polysaccharides? These polysaccharides are also known as glycans. Okay, these are considered to be glycans. Okay, they are made up of repeated units of monosaccharide, not just one, two, or three. Repeated. Cellulose na many glucose subunits, starch also many glucose units. Okay, so repeated units of monosaccharides, again by which type of bonds? Disaccharide also glycosidic bonds, polysaccharide is also glycosidic bonds. Glycosidic bond is formed by removing one water molecule. Okay, during its formation, water molecule is released at each condensation. This helps the reducing the bulk, making it almost insoluble. This is the reason why polysaccharides are considered to be soluble or insoluble. Insoluble. Okay, and decreasing its effect on the water potential and the osmotic potential of the cell. Unlike sugars, they are never considered to be sweet. Polysaccharides are sugar அப்படின்னு கேட்கலாமா சொல்லலாமா சொல்லக்கூடாது so these are never considered to be sweet, and they are never called sugars. They are polysaccharides. Okay, not sweet. They are ideal as storage and structural component. Ideal for two purposes: storage or structural component. They are of two types: homoglycans, heteroglycans. They are not called sugars, they are called glycans. Two types: homo, hetero. Homoglycans made only one type of monosaccharide. If it is containing glucose, continuously glucose, okay, that is homoglycans. If it is containing condensation of two or more types of monosaccharide, it is considered a heteroglycan. You can see examples: starch, glycogen, cellulose. Glucose is made up of glucose, fructose is made up of fructose, and galactan is made up of galactose. These are examples of what? Homoglycans. Okay. Heteroglycans are examples: hyaluronic acid, agar, agar which is used as a nutritive medium, right, to grow microorganisms like bacteria. Chitin, chitin is the cell wall material of what? Fungus, exoskeleton of cockroaches. Peptidoglycan, cell wall material of bacteria, right? Peptidoglycan plus teichoic acid, gram positive, gram negative, we discussed. Moreover, it is also known as N-acetyl uramic acid. It is bacteria, fungi, cockroach. Okay, these are heteroglycans. Easy? Okay. Now we'll move on to the next. You guys can note it again. Don't worry, I'll send the notes to you. Okay. We'll move to the next. Sorry. Storage polysaccharides. Polysaccharides which are meant for storage, not for structural thing. Okay. So starch. Starch edhula irukku? Potato we have seen, right? It consists of starch, right? Okay. So carbohydrate in the form of reserve material for plants and is a dietary source for animals. Okay. It is a repeated unit of glucose. Starch is amylose plus amylopectin polysaccharide component. Okay. So glycogen is again a storage polysaccharide. Carbohydrate reserve in animals, hence referred to as animal starch. Carbohydrate reserve in plants, starch. Carbohydrate reserve in animals, glycogen. Okay, glycogen. Again, it is a glucose as the monomeric unit. Okay, inulin. It is a polymer of not glucose, a polymer of fructose. It is also known as fructosan, found in dahlia, a type of a plant. Bulbs, onion. We have onion, garlic. Okay, then easily soluble in water. This is actually soluble in water, but is a form of a polysaccharide. Generally, polysaccharides do not dissolve, exception. Okay, sorry. Inulin is not readily metabolized in the human body and is readily filtered through kidney. It cannot be metabolized. It cannot be used. That is why kidney filters it out. Okay, through excretion. Hence, used for testing kidney function. GFR. GFR la எது யூஸ் பண்ணுவாங்க? Which they use? கிட்னி ஃபங்க்ஷன் ஆகுதா இல்லையான்னு செக் பண்றதுக்கு எது யூஸ் பண்ணுவாங்க? இன்யூலின் இருக்கா இல்லையான்னு செக் பண்ணுவாங்க. யூரின்ல யூரின் டெஸ்ட் பண்ணி, சரிங்களா? So these are considered to be what? Storage. Structural, cellulose, cell wall material. Chitin, cell wall material. Okay, cellulose again, it is totally absent in animals. Most abundant organic substance in plant kingdom. Most abundant. Note this, because every plant kingdom is containing primary cell wall component. Okay, sorry. Second most abundant organic substance, kind. First, second organic substance. Okay, va? Sorry. Heteropolysaccharide type found in the exoskeleton of some invertebrates like insects as well as crustaceans like prawns. Okay, prawns, crabs, etc. As well as provides both strength and elasticity, becomes hard when impregnated with calcium carbonate. Okay. So, okay. So by this we have one more topic here is reducing versus the non-reducing sugar. Okay, reducing, non-reducing. Sugars that can reduce metal ions using which test? Benedict test or Felling's test. These are tests. Okay, due to free aldehyde or ketone groups. Suppose a polysaccharide, repeated unit of what? Glucose or whatever. If at one end it is having a free aldehyde or a free keto group, then it is considered to be a reducing sugar. Okay, example glucose. Not only polysaccharide, monosaccharides. Okay, specifically applies to monosaccharides. Glucose, fructose, maltose, lactose. Okay. Lactose is a disaccharide. Okay. So now non-reducing sugar, they do not have free aldehyde or ketone group, so they cannot react with what? They cannot reduce metal ions. Eventually, they are considered to be non-reducing sugars, and because they are involved in what? Glycosidic bonding. Sucrose, since glucose, one of the aldehyde group and one of the ketone group are involved in bonding by releasing the H2O molecule. So that's why this is not considered to be a reducing sugar, and it is a non-reducing. When we'll know whether a given sugar is reducing or non-reducing? By doing which type of test? Felling's test or Benedict's test. Okay. So reducing sugars play roles in metabolism, diagnostics. Non-reducing sugars like sucrose serve as transport and storage forms of carbohydrate. Got it? Uh, so reducing ends in polysaccharides like starch, glycogen. One end has free anomeric carbon, that is at carbon 1 in starch. Okay, what is that? There is a free that making it a reducing end. So polysaccharides might have one of the end as reducing and the other end might be a non. I'm telling might be. It may be or it may be not. Okay. So non-reducing end, the other end of the chain is bound by a glycosidic linkage. Wherever the linkage is there, so there is no free anomeric carbon. This makes it non-reducing. Enzymes like amylase enzyme act from the non-reducing ends. If amylase is what? Salivary amylase is found in the saliva. It is responsible for breaking down what? Starch, right? So if it is going to break down starch, it will start its action from which end of the starch? Reducing or non-reducing? Non-reducing end, which is having the glycosidic bond. So it will start from the glycosidic bond and it will eventually go to the last where it is free. Okay, understood? Amylase enzyme. So this is the thing, and at the end we'll discuss some questions. Identify the substance having glycosidic bond and peptide bond. Peptide bond is a bond which is seen in proteins. Okay, we have not discussed it, but it was a question which was asked in NEET 2020 question paper. Okay, whether it is glycerol or trypsin or cellulose or lecithin, inulin, which one? Inulin. Okay, inulin is a carbohydrate polymer, has a glycosidic bond, and insulin is a protein hormone. The question was not actually aligned properly. It has a pair. Inulin is a carbohydrate polymer. Insulin is a protein hormone. Okay, so it has what? A peptide bond. Okay. Next, the second question: sucrose on hydrolysis produces. Sucrose is consisting what? Fructose. So it will produce what? Alpha D glucose and beta D fructose. Again, alpha means what? The OH group at the C1 is facing upwards or downwards? Beta means upwards. D form means left or right? Right. Which is right? The alcohol group, again which carbon? Fifth carbon, C5. Okay. Now, which of the following is a non-reducing carbohydrate? Maltose, sucrose, lactose, ribose 5-phosphate. Sucrose. Okay. Why? Both the end, fructose as well as glucose. No free ends for aldehyde and ketone. Which of the following are not secondary metabolites in plant? Morphine, codeine, amino acid, glucose, vinblastine, curcumin. Did we discuss this? But these are secondary metabolites. You have to note this. Curcumin is found in what? Curcumin edhula irukum paathambi? Curcuma longa. What that is? Curcuma longa is the scientific name or the binomial name of which plant? என்னப்பா இது கூட தெரியல? பாட்னி படிக்கிறீங்களா நீங்களா? This is Turmeric, right? Huh? So curcumin is found in what? Turmeric. Curcumin இப்போ மெடிசனுக்கு யூஸ் பண்றாங்க. மெடிசின் லைன்ல தானே இருக்கீங்க? curcumin சரிங்களா? எதுல இருக்கும்? டர்மரிக்ல இருக்கும். செகண்டரி மெட்டபாலைட்டா இல்லையா? நீங்க வரல. செகண்டரி மெட்டபாலைட். சரி. Vinblastine is also a secondary metabolite. Okay. Rubber and gum, second metabolite. We have this. These are extra points. We have to note this. Okay. Vinblastine, curcumin, and amino acid and glucose is the answer because it is not a secondary metabolite. It is a primary metabolite. Okay. So by this we have completed our biomolecules part 1. Next part la enna paapom thambi? Proteins. Proteins முழுசா சொன்னா, when we talk protein as a whole, do we tell it as macromolecule or micromolecule? Macromolecule. But when we say amino acid, micromolecule. So in the next class we'll be dealing with amino acid and protein. I hope the class was okay. Now we'll move on to the next class. Okay.